the inborn errors of metabolism. This is one more type of metabolism. After completing, I'm left with only this small chapter, and then we will talk about developmental biology over there. Developmental biology also is a very important topic over here. Um, Coming to inborn errors of metabolism, we will talk about what exactly is the errors that is occurring during metabolism, and also what are the different types of genetic disorders that you get because of this metabolic errors. We have to credit this topic to Archibald Garrett, 1908, who had uh, described about this inborn errors of metabolism. So what exactly is this inborn errors of metabolism? So something like we eat a food for energy. We either eat them in the form of carbohydrates, proteins, and lipids. So that has to be broken down or metabolized to the next product, and this next product will be metabolized to smaller products. The complex food particles have to be metabolized to simpler food particles so that the blood can assimilate them and they can give us energy to different parts of the cell. What happens over here? Each pathway needs a specific enzyme to break them down. And if that enzyme has some problems, if the production of the enzyme has a problem over there because of the gene mutations or gene deletions over there, then you're not able to produce that enzyme. And hence, the conversion from complex particles to simpler particles is stopped over there. So that is called as inborn errors of metabolism. So exactly what they really mean over here is the inborn errors of metabolism are referred to congenital metabolism. That is, congenital metabolism is where they have some problem in the childhood where the metabolic pathways is not clearly done. Or this can also be inherited metabolic disorders. And mostly they are autosomal recessive disorder. We talked about autosomal dominant disorder. We talked about autosomal recessive disorder. Among this, autosomal recessive disorder means both the parents should carry this particular gene that is having some problem over there. So the chromosome, which is not having this particular gene to produce that enzyme, should be present in both the parents' chromosome over there. Only then that can be inherited to the children. That is called as autosomal recessive disorder. We talked about this autosomal recessive disorders, or the genetic disorders in the previous topic over there. These diseases involve failure of the metabolic pathways involved in the either breaking down or storage of the carbohydrates, fats, and proteins due to enzyme defect. I told you enzyme is the one that needs to be needed to break down from one complex particle to the next simpler forms over there. Although this is very rare, 1 in 2,500 children will be having this problem over this, as this is an autosomal recessive disorder. And this is always because of defecting the single gene that codes for that enzyme that needs to convert from one product to another product. And these metabolic disturbance, if left untreated, can result in cognitive impairment. That is, the development of the brain is not properly done, and sometimes organ failure, and even death in the children over there whoever is having this gene that is present in the community. They can be present at any age, and therefore awareness of this disease is needed, and the evaluation is critical for management of inborn errors of metabolism. Now, this inborn error of metabolism can also be identified by some tests that can be done. Neonatal test is done over there. I'll be discussing about this topic in the end. Neonatal test can be done to find out if there's any inborn errors of metabolism in the growth of the fetus in the stomach, in the womb. Okay, now coming to inborn errors of metabolism, as I told you, will be eating proteins, carbohydrates, and fats in our food. That is, carbohydrates in the form of rice, proteins in the form of dal, meat, and lipids in the form of oil, ghee, butter, and so on and so forth. Now, these all have to be broken down into simpler products from the complex nature to simpler nature, so that is broken down up till pyruvate and acetyl-CoA. Glycogen are the carbohydrates broken down to pyruvate and acetyl-CoA. Protein is also broken down to pyruvate and acetyl-CoA. Lipids is also broken down to acetyl-coenzyme A. All these undergoes another cycle, that is the Krebs cycle or TCA cycle, citric acid cycle, which is all done in the mitochondria over there. And in the mitochondria, why this has to be again going through this metabolic pathway is to produce ATP. Why we eat food? We eat food to get energy. Energy is produced by the mitochondria from the food products that has been broken down to acetyl-CoA and pyruvate, which is taken care of in the CCA cycle over there. Now, if there is any problem in this breaking down, then the child or elders or adults will be having some problems over here. So here we have three different types of food, but we'll be concentrating in this particular class, we'll be concentrating on amino acid metabolism, that is protein metabolism to amino acids. So when there is a problem in this amino acid metabolism, then the child can be having these different types of inborn errors of metabolism, that is phenylketonuria, tyrosinosis, albinism, alkaptonuria, and goitrous rheumatism. So what are these? We'll be talking about that then one by one over there. Okay, now coming to phenylalanine over there. Now, the proteins that are going to be broken down, a phenylalanine is an essential amino acid. So the amino acids are of 20 to 21 types. So under this amino acids, you have two different types, essential amino acids, non-essential amino acids. Essential amino acid, that is you need to eat so that you provide those amino acids in your system. And non-essential amino acid is the one that your body is already going to produce from the essential amino acids that we take in, right? So you've understood this. You take the essential amino acid to also produce non-essential amino acids in your body over there. So you have 9 essential amino acids and 11 non-essential amino acids over here. So among that essential amino acid, phenylalanine is one among them, which is very important, which is found in protein-rich diet like meat and dairy farm. Dairy product, that is milk, cheese, paneer, and so on and so forth. Why you need this? This is very much crucial to produce dopamine and tyrosine. What is dopamine? Dopamine is a neurotransmitter which is going to give you a feel good factor to your body. So, for example, when you write an exam and you get good marks, you're going to feel that, yeah, I'm going to be rewarded. That is the feel good factor. Yes, that I've achieved something. That feel good factor is produced because of this dopamine. Not only that, when a child is waiting for its parents, thinking that a parent is going to get a chocolate, now that is a feel good factor. So they keep waiting until the parents come so that they can have the chocolate in their hands. Now that is a neurotransmitter over there. That is dopamine. Now tyrosine is also a amino acid, but it is a non-essential amino acid or conditional essential amino acid. Now, what is the function of this non-essential amino acid tyrosine? Oh, that's the next one, tyrosine. The phenylalanine is metabolized to tyrosine. The tyrosine is the one that gives cognitive alertness. That is, when you're alert, say for example, when something is moving, immediately you look over there, or there's some sound coming outside, you're going to react for that sound. Cognitive alertness is because of tyrosine. Then also stress, when there is a stress, how you decode that stress. Then also how the thyroid function is occurring, all this is because of tyrosine video. So I'll just quickly make you understand what this paper is all about, like over here, the slide over here. Phenylalanine has to be metabolized to tyrosine with the help of an enzyme called phenylalanine hydroxylase. Now, this enzyme is very, very important over here to break down phenylalanine to tyrosine. If this is not present, then the phenylalanine cannot be metabolized to tyrosine. So, this enzyme is very much necessary. All those that ends with an ACE is called as an enzyme. I don't have to tell you all that, but still. Now, tyrosine that has been metabolized, from phenylalanine has to be further metabolized to 4-hydroxyphenyl pyruvic acid with the help of what enzyme? Tyrosine aminotransferase, right? Now, this 4-hydroxyphenyl pyruvic acid has to be again metabolized to homogenesic acid with the help of hydroxyphenyl pyruvic acid dioxygenase enzyme. Now, with this help of this enzyme, this 4-hydroxyphenyl pyruvic acid will be broken down to homogenesic acid. Homogenesic acid will be further metabolized to mallyl ester acid with the help of homogenesic acid dioxygenase. And then this acetic acid that has been metabolized will be further metabolized to fumaryl astatic acid and further to fumaric acid and astatic acid with the help of hydrolase enzyme. And this astatic acid will undergo to metabolize to form acetyl-CoA, which then enters into the TCA cycle to produce the different forms of metabolites. That is, either you need a TCA, the ATPs for production of energy, or some neurotransmitters that is needed. All the thyroid hormones that are to be treated. All these are to be going to be brought about by this breaking down of this metabolic pathway. Now, what is the main important points over here? Now, phenylalanine has to be metabolized to tyrosine, we told, with the help of what enzyme? Phenylalanine hydroxylase. Now, if this enzyme is not produced by the gene, for example, if that particular gene that is present on the chromosome has undergone some mutations, has lost its activity, or deletion has occurred, then what happens to that? When this enzyme is not present, then phenylalanine will not be broken down to tyrosine. And if this is not going to be broken down to tyrosine, phenylalanine accumulation occurs that leads to an inborn error called phenylketonuria. That is what we are going to talk about. The first example over here is phenylketonuria. So what I told you, phenylketonuria is because of the phenylalanine not being broken down or metabolized to tyrosine with the help of hydroxylase. Right? This hydroxylase enzyme is not produced because of some genetic disorder or gene that has been mutated. Now, this is present. This particular enzyme is the gene that is support for this enzyme is present on chromosome number 12. So you can see over here 12q23.2. What does that mean? Chromosome number 12, which arm? Q arm, which position? 23.2. So this is the place where PAH is going to be produced. What is PAH? Phenylalanine hydroxylase enzyme. Because this enzyme is not being produced because of the gene that is being mutated, now you're not able to break down or metabolize phenylalanine to tyrosine, and that leads to inborn error for phenylketonuria. Now, what is expression symptoms that you have when phenylketonuria is excess over here? Growth failure occurs, poor skin pigmentation, microcephaly, small head is called as microcephaly, global development delay, and severe intellectual impairment. IQ at 65 over there. So normal IQ is at 70 to 150, but here IQ is 65 over there. But however, because of this newborn screening programs that we have, there are different types of newborn screening programs with early detection. Children with PKU can now expect to lead a normal life over here, provided the parent will undergo this newborn screening programs over there. Now, coming to the next one, thyrosinosis. Now, what is this tyrosinosis? Now, we talked about phenylalanine being metabolized to tyrosine with the help of hydroxylase. Fine. There is no phenylketonuria because they have hydroxylase. But this tyrosine have to be metabolized to 4-hydroxyphenylpyruvic acid. With what enzyme? With the enzyme tyrosine aminotransferase. Now, this tyrosine aminotransferase, if this is not being produced, then this leads to accumulation of tyrosine, and this leads to tyrosinosis. So everything has to be cleaned. Something like when the garbage is getting filled up, it has to be cleaned. Otherwise, what will happen? They will create some problems at home. So in the same way, if there's anything excess in our body, that will create some problem. And this is called as tyrosinosis, where tyrosine is not being metabolized to 4-hydroxyphenylpyruvic acid. Now, this problem is found on which chromosome? Chromosome number 16, Q22.2 to Q24.3. So that much amount of gene is mutated over there, and hence this tyrosine aminotransferase is not produced over there. Now, symptoms of untreated tyrosinosis leads to liver failure, right? Because excess of tyrosinosis accumulates over there. And tyrosine, which is a non-essential amino acid, is involved in the synthesis of epinephrine, norepinephrine. These are the hormone, the neurotransmitters, which is going to keep yourself healthy. Otherwise, what will happen if there is less amount of this neurotransmitters? Then the person will undergo depression, ADH, attention deficit disorders, headache. Frequent headache is because of this tyrosine not present over there. The dopamine, of course, the feel-good factors, thyroid hormones, and pigment melanin. The color of the skin is because of this pigment melanin. And all this goes for a toss if tyrosine is not broken down to 4-hydroxyphenylpyruvic acid. And this also Screening can be done so that early intervention can lead a better life, and the doctors can also prescribe low protein diet because they are not able to break down the protein. So low protein diet can be given, and some other protein formulas can be given so that the child stays healthy and live its normal life. The next one is alkaptonuria. Now we have talked about phenylalanine to tyrosine, tyrosine to 4-hydroxyphenyl pyruvic acid. A 4-hydroxyphenyl pyruvic acid is broken down to homogentisic acid. Homogentisic acid has to be broken down to mallyl acetoacetic acid with the help of homogentisic acid dioxygenase. Now, if this enzyme is not present, then this leads to alkaptonuria in the system or in the body. Now, this is again an autosomal recessive. All these are autosomal recessive disorders wherein both the parents have to have this to be passed on from the parent to children. Now, this is present on chromosome number 3q13.33. 3q3 is chromosome number 3. Q is the Q arm, which position 13.3 is a position where this particular gene is present to produce what? Homogentisic acid dioxygenase. Now, conversion of the homogentisic acid to malolactic acid is being blocked. Why? Because this enzyme is not there. That leads to alkaptonuria. Now, accumulation of this homogentisic acid in the body leads to multiple systemic diseases like the urine turns dark. So when you fill up the urine and leave it outside, when it is exposed, you can see in the picture over here, this is a normal urine color over here. But those people who are affected with this alkaptonuria, their urine turns dark color when exposed to sunlight. And also, they also have this ochronosis where the soft pigments, connective tissues, becomes a little bit darker in color. You can see in the ear, the dark color in the ear. This was because of ochronosis, the accumulation of alkaptonuria, the homogentisic acid in the system over there. This can also lead to osteoarthropathy, that is the large bones, joints problem. Kidney stones can also occur because of this bone fracture, frequent bone fractures, ligament and muscle ruptures is all because of this homogentisic acid accumulation over this. So people with this alkaptonuria have disabling symptoms such as pain, poor sleep, breathing symptoms. And by the time they turn 50 to 55 years, and because of this osteoarthropathy, they might have to undergo some joint replacement surgery because they are having this problem, that is accumulation of HGA in their system. The next one is albinism. Now, when you're talking about albinism over here, I want you to understand this picture over there. Till now, what we talked about, phenylalanine turning into metabolized to tyrosine, tyrosine metabolized to homogentisic acid and malolactic acid. Now, this tyrosine has to also undergo two types of metabolism over here. Now, when this tyrosine has to be metabolized to DOPA, that is dihydroxyphenylalanine over there. Now, this DOPA is going to produce melanin, that is pigmentation in our body. Now, if this is not broken down properly, then that leads to albinism. That is, you must have seen a person is completely white in color. That is, there is no color at all, completely white color. You must have also seen, you must also talk about rats that are white in color, albino rat and all those type mice. So in the same way, because of the melanin is not produced in the skin, then these people are affected with albinism over there. Now this is the picture of albinism people, where you have, this is also an autosomal recessive genetic disorder over here, caused due to the mutation of tyrosinase gene over here. Now this tyrosine has to be broken down to DOPA, that is dihydroxyphenylalanine, DOPA, dihydroxyphenylalanine. Now, from tyrosine, tyrosine has to be metabolized to DOPA with the help of tyrosine hydroxylase. Now, this is the enzyme, tyrosine hydroxylase. Now, this is present on chromosome 11, which arm, Q arm, and which position 14. Now, because there is a gene mutation in this particular band over there onto the chromosome Q14, now this leads to mutation of the gene, tyrosinase gene, that is not going to be produced over here. Now, this tyrosine hydroxylase normally hydroxylates the tyrosine to dihydroxyphenylalanine, and this phenylalanine can be further broken down to melanin. Now, because this is not being done from DOPA to melanin, albinism occurs. So here, what happens is because melanin is not produced, you can find these people skin is very much white in color. There is no pigment at all, no color at all, pigmentation. So these people are at a risk of affected with cancer, skin cancer, because there is no melanin in the body, and the melanin is the one that is going to protect us from UV radiation. So subjects have to avoid excess UV light exposure. Direct sunlight is not recommended for them, and they have to have lenses which has UV protection. Bifocals and low vision aids may also be considered because such people cannot see long distance clearly. They are not able to clearly see the objects easily. The irritation occurs in them, and they are not able to concentrate. The cognitive alertness is a little bit less in them. So this is albinism over there. And the last one is goitrous crinatism over here. What is this goitrous crinatism? Now, this is an autosomal recessive condition again, and goiter is present in them. You must have heard of that goiter, where thyroid problem, the iodine problem, wherever who is having this iodine deficiency, they have this goiter. In the same way, here again, this is an autosomal recessive condition where uh, the goiter is present because the tyrosine over here. Now, because when the tyrosine is metabolized to DOPA, DOPA gives melanin, and so the patient is not having albinism. In the same way, this tyrosine has to be metabolized to thyroxine. Tyrosine has to be metabolized to thyroxine with the help of thyroid peroxidase enzyme. TPO is a thyroid peroxidase enzyme. Now, this helps in metabolizing tyrosine to thyroxine. This thyroxine, that is T4, and also this is also broken down to triiodothyroxine, that is T3. Now, these are blocked if you don't have thyroxine, the peroxidase enzyme over there, which is thyroid peroxidase enzyme, which is going to break down tyrosine to thyroxine. So, what is the problem that a child is having over here? Congenital cranitism with a severe hypothyroidism. Thyroid is not produced in them, so it's called as hypothyroidism. That can lead to mental disorders and also the swollen faces that you can see over there. Different types of problems can occur. Mental retardation also is one of the important points over there when untreated over here. So for all these things, what is most important is here this is what I was talking about, methods of prenatal diagnosis. Now, why is diagnosis important over here? Now, this diagnosis is important for parents who are aged completely. So, for example, from the age of 22 to 30 years, this is the prime age where all the gametes are normal and there is no problem with the children that are born to such parents over there. Why? Because the eggs are young, the sperms are also you don't have much mutations in them, so everything is normal. The child that they have from these parents are normal. But now, because of the conditions unknown or you cannot express, it can be career orientations or it can be some other important projects that has to be completed. Now parents are trying to delay their childbirth. So, for example, about 30 years, 35, 40 years, 45 years, the eggs are aging over there. As the eggs are aging, genetic disorders can also age. Now, having said that, you cannot blame only the eggs that are going to be aged. Even the sperms has its own problems over there. The sperms, as a man is aging, the sperms also has increased mutations in them. Why? Because every day the cell have to undergo divisions, mitotic division, mitosis and meiosis divisions over there. Now, the more amount of mitosis and meiosis, the more amount of mutations that can occur, chances of mutations that can occur. So even the sperms can have some mutations in them. So to avoid all those things, even though when these parents want to have children of their own, then they can undergo this methods of prenatal diagnosis. These are of two types: invasive and non-invasive type. Under invasive amniocentesis, that is when the amniotic fluid is taken from the womb, then that is called as amniocentesis. Cordocentesis, when the umbilical cord, from the umbilical cord, you can take out that blood, so that is called as cordocentesis. You can see the cordocentesis over there. Then chorionic villus sampling, the chorionic villi that is present near the placenta, even the blood can be taken from there for checking out the child's DNA. Biopsy from fetal tissue can also be done if the doctor sense there's some problem in the fetal production, fetal development, then biopsy from the fetal tissue. This has to be very, very carefully done. Not all doctors can do that. Specialists can do that. Then celosynthesis. All these are different types where you can do this particular samplings over there to find out what is the karyotyping of the child. Then non-invasive technologies, ultrasonography, there is a scanning that can be done to find out if the limbs and the growth is properly occurring or not every trimester. MRI scanning can also be done. Cell-free fetal DNA. Now, this is cell-free fetal DNA, the DNA that can be collected from the mother and checked out for any problems of the fetus can be done. And triple tests, there are different tests that can be done. Then also fetoscopy can also be done. So that's a fetoscopy between 3 to 20 weeks, where a catheter is induced into the womb. You can see a small catheter over there. It is inserted inside to check out the live movement of the child and to find out if there's any problem in if there is any amniotic band. In the last class, I was talking about the amniotic band, the amniotic tissue that is going to get tied up, or umbilical cord, or even twins if they are going to be very, very much close together. All this can be checked with this fetoscopy over there. So when you do this prenatal diagnosis, you can come to know that if there's any Down syndrome, Pator syndrome, or Edwards syndrome, within the specific period of time, the parent can undergo abortions, or otherwise what happens if that becomes, if the child is born with such ailments, it is a problem for the parents as well as for the child over there. So that is prenatal diagnosis that can be done. So this much of amount I could do for the unit one. So now we'll go to the next one, developmental biology, because this is also a very important topic over here. Under developmental biology, the first one we'll be talking about is gametogenesis over here. So what is gametogenesis? Gametes, formation of gametes is called gametogenesis. That is the formation of the sperms and ovum is called as gametogenesis. Then under that, we'll be talking about fertilization and types of cleavages of the zygote over there. So under this gametogenesis, the first one what we'll talk about. First, before going into the topic in detail, we'll first understand what is developmental biology and why it is so important as a separate subject over here. Now, growing field of stem cell research. You must have heard of stem cell research a lot. Stem cells are stems, not the stems of the plant, but the stem cells that you have within us, the adult stem cells, the gastrulation, the blastula cells that is found over there. All these cells are totipotent, some are pluripotent, some are oligopotent, and all those things. Now, these have a specific signal to grow into an organ what you want. So that is called a stem cell research. Now, this ranges from basic research to clinical applications by using embryonic stem cells. Embryo stem cells are more of pluripotent, and they have the ability to grow into whatever you want them to grow, provided you give them the growth factors over there. And also you have this mesenchymal stem cells and also induced stem cells over there. So there are some problems which can be cured by these stem cells over there. So development biology helps in that. Embryos of various species like mice, chicken, and zebrafish, the small fish, zebrafish, are often used to investigate genetic, physiological, and morphological, pharmacological influences on organogenesis or disease progression here. Now, what is this genetic, physiological, and pharmacological? Pharmacological is you can also test. So you're preparing some new drugs over there. You want to use them on the stem cells, the embryonic cells over there, so that you want to know what is the effect that it will have on the adults over here by using this mice, chicken, and zebra. Why mice, chicken, and zebra? All these mimic a little bit about the human genome also. So that is the reason why we use mice, chicken, and zebra embryos for this various experiments. Embryonic models are widely used in treatment of cardiovascular research because nowadays, drop of the hat, you find everyone having some cardiac problem, or somebody is having heart attack, somebody is having um, the clot in the blood, angioplasty has to be done, stent has to be used. All these problems are seen over here. So to treat that also, embryonic models are used to treat this particular diseases. And also there are various development process which helps in revealing mysterious fundamental process like regeneration, metamorphosis, and teratogenesis. Regeneration. You must have studied about this regenerations where we talk about how the starfish can regenerate if the limbs are broken. The planarians can regenerate. We also have an organ that can regenerate, that is the liver. But why not other part of our body? Why can't they regenerate? So research is going on that metamorphosis, how metamorphosis occurs, and teratogenesis only. So various developmental process which help to develop new drugs and therapies for the prophylaxis of various disorders, that is lessening the pain of the various disorders. And the study of development biology plays a crucial role in reducing the number of neonatal deaths. So nowadays we have a lot of abortions, or the moment the child is born, they die. So all these can be addressed when we have this developmental biology studies, so that we can reduce the number of death over there. So that is the importance of developmental biology. To understand this research, first of all, we should understand what are the cells that we have, how they are produced, and what are the various functions that they do. Only then we can use that into the research over here. So for that, we are going to talk about that one by one in detail over here. Now coming to gametogenesis over there. So under gametogenesis, we are going to, genesis is to give birth to gametes. How the gametes are produced is called as gametogenesis. Under this, we are talking about spermatogenesis and oogenesis, production of sperm cell, production of ovum or matured egg over here. Now, during meiosis or meiosis, two cells divide, separate from the paired chromosome in the nucleus, and you get you make them separated to form haploid number of chromosomes. From diploid to n, from 46, you're going to have 23 pairs, 23 each over there. So that is called as meiosis, or reduction cell division is called as meiosis. So when you have this reduction division, only then you're going to get this gametogenesis that is going to occur in the spermatogenesis, oogenesis. So during spermatogenesis, for example, if you're talking about males over here, male have 46 XY, right? X chromosome and Y chromosome. Now this has to get separated. How do they get separated? 23 and 1X, 23 and 1Y. So that is the reaction division. 23X, 23 Y chromosome and 23 autosome, 23 autosome and 1 X chromosome. In the females, you have 2 X chromosome and 46 number of chromosomes. Then this 46 has to divide into 23, 23, and each ovum is going to have 1X and 1X over here. So you are reducing it from 46 to 23. That is 2N. 46 is 2N. You are reducing it to haploid, that is 1N, that is 23, 23. So this is what is called as reduction division during meiosis over here. So how are these produced? That is what we are going to talk about over here. So if you can see in this picture over here, during meiosis, in the interphase stage, homologous chromosome pair, that is the prophase one of that the different stages that I talked about, leptotin, zygotin, and all those undergoes. And this undergoes meiosis 1 over here, reducing the number of chromosomes to haploid number of chromosomes over here. And this haploid number of chromosome has to undergo another type of division, that is meiosis 2 over here, to reduce into single gametes over there, single chromosomes over there. And so you have this haploid number of chromosomes. So from one mother cell, you're going to have 4 cells that is in the sperm. And in eggs, from one mother egg, you only end up having one matured egg over there. Why? We'll be discussing about all this. Now coming to spermatogenesis, how the sperms are produced. Now, production of the sperm cells is called spermatogenesis, and it takes place in the seminiferous tubules in the testis. Now, if you can see in this picture over there, so that is the testis over here, where you have The seminiferous tubules, that is, they're all coiled tube-like structures. So you can clearly see in this, coiled tube-like structures is called seminiferous tubules. And this is covered by tunica albuginea. And that is a testicular lobules that are present, and these are divided by the septum in between. So these are the septums that you find over there. And all those coiled tube-like structures are called as seminiferous tubules. And in this seminiferous tubules is where the sperms are produced. Now, if I can take a piece of it and cut it open and show the transverse section of it, when the tube is cut open, so you have this tube. When I cut open and show this part over there, so you have this cells over there, the mother cells, the primordial germ cells at the edges of the tubules over there. And as you move inside, there is a lacuna that is formed over there where the sperms get accumulate over there, and from there they are put into epididymis, and from epididymis they are moved to vas deferens and outside over there. Now, before that, you will have to understand what are these cells that are present over here. If you can see in this picture, a little bit of magnified part of this portion. When you magnify them, and if I show them over here, now this is the germinal cell over there, which is a diploid number of chromosomes that are present. That is 46 number of chromosomes that are present. In between are Sertoli cells. These are transferred by the Sertoli cells in between. Now, this cell has to undergo mitosis to grow in number of cells, and this leads to production of primary spermatocytes. Primary spermatocyte undergoes meiosis 1 to produce secondary spermatocytes. That's the secondary spermatocytes. Now, this secondary spermatocytes undergoes meiosis 2 to produce spermatids. The round ones are the spermatids, and this spermatids will develop into sperms over there. So this is how they occur over there. And what are the stages over here for the spermatogenesis to occur? There are 3 stages: proliferative phase, mitosis, meiosis phase, and post-meiotic phase. That is a maturation phase that is going to occur over here. So here, what you're going to see is proliferative phase. Now, what is proliferative phase? Proliferative phase is where they have to increase the number, and that is called as proliferative. So you have one mother cell, the germ cell or the mother cell over there. This mother cell has to keep producing sperms, but this has to undergo multiplication. Why? Because you have only less amount of them. Now this has to undergo mitosis to increase in number. Some are kept in memory cells, some undergoes the divisions of meiosis over there. So proliferative phase, which involves the rapid multiplication of primary germ cells. So these are the primary germ cells that are present at the tubular end of them, which begins to with the arrival of the primordial germ cells. And these primordial germ cells get incorporated into the cords, that is the seminiferous tubule cords over there. What is this primordial germ cells is? Primordial germ cells is the one during the development of the fetus. The cells get accumulated on different parts of the body, but some cells from the yolk sac, they move into the gonadal region over there. And those cells that goes to the gonadal region are called the primordial germ cells. So these are the primordial germ cells. And this primordial germ cells, some of them, when they move to the gonadal regions, they get accumulated in the seminiferous tubules. So this is the seminiferous tubules over there. And from here, they get differentiated into spermatogonia. Now, how are they going to go? Well, this spermatogonia resides in the stem cell niches at the junction of the Sertoli cells. So these yellow color ones are the Sertoli cells that I was talking about. There are some more important cells. This is the Leydig cells over there. This is the Sertoli cells. And there are some blood vessels that are interspersed in between. The blood vessels supplies oxygen. The Sertoli cells nourishes the developing spermatogonia. And the Leydig cells are the one that produces the testosterone hormone in the males over there. The Leydig cells produces a hormone called as testosterone. Okay. Now, the spermatogonia joined to the Sertoli cells. Why? Because they have to be nourished for each cell division that has to occur. So, Sertoli cell is also important over here during the process of development. Now, that is proliferative phase. That is when this primordial germ cells will undergo mitosis. During this mitosis, nourishment has to be given for production of primary spermatocytes over there. So this Sertoli cell is going to take care of all this. That is proliferative phase. The next one is the meiosis stage over there, meiotic phase over here. The undifferentiated spermatogonia undergoes mitotic division to produce primary spermatocytes. So this is proliferative phase. The spermatogonia undergoes mitosis to produce primary spermatocytes. Now, this primary spermatocytes which has been produced will have to undergo meiosis over here, meiotic divisions over there, meiosis, or simple word meiosis over there, meiosis or meiosis, one and the same. So, meiosis has to be undergone over here for regulation of the primary spermatocyte to secondary spermatocytes over here. So, what is occurring over here? Now, in this picture, if I have to show you all, proliferative phase, mitosis occurs. Now, after mitosis, the spermatogonia is going to become primary spermatocytes. They are still in diploid number of chromosomes, that is 46 chromosomes are present. Now, this 46 chromosome has to undergo meiosis 1 division. Meiosis, remember, two divisions have been meiosis 1 and meiosis 2. They have to undergo meiosis 2 division over here. After the meiosis, they divide into two cells, and these two cells are called secondary spermatocytes. That is 46 have become the haploid number of chromosomes. And this have to undergo another meiotic division over there, meiosis 2 over there. From here, this will divide into 23x, 23y, 23x, 23y. Because why? This is still having xy, this is also having xy. Now, X and Y has to get separated into two different gametes over here. So that is the reason why they have to undergo the second division of meiosis over here. And once the meiosis occurs, one cell becomes two and this cell becomes two over here. So from one single cell of primary spermatocytes, you have four cells over there. That is it. Testis over here. For the sperms to develop, from one cell, you have four sperms over here. Now, this is called as the spermatids. Now, these spermatids are still immature, and with the help of Sertoli cells, the nutrition is given to them, and this will become a functional matured sperm over there. So, this is the meiotic phase over there, and this is a maturation phase. How the maturation phase? The spermatids, which have been produced, will have to mature itself to become a sperm over there. That is called spermiogenesis over there. That is a maturation phase over here. Now, you have to understand the shape of the sperm over here, how from spermatid they're going to become a mature sperm over here. So there's also different stages over here, and changes during spermiogenesis or maturation sperm is about 200 million per day. So per day, a man can produce 200 million sperms, which has to be produced on a daily basis over there. And mitotic and meiotic division undergoes over here. Now, because this mitosis and meiosis undergoes on a daily basis, that is what I told you, mutations can increase as a man is aging over there. Now, first we'll talk about how the maturation is occurring over here. So you have a spermatid over here. What is a spermatid over there? Now, this is the spermatid that has been produced from the secondary meiotic division or the second meiotic division. You have the spermatids over there. Now, these are having 23x, 23y, 23x, 23y. Although they are all attached over there, only after maturation, they get separated to form the mature sperm over there. Now, this spermatid that is being formed will have 23 chromosomes, either X or a Y. This is also having a Golgi apparatus. Now, this Golgi apparatus goes and settles at the head region of the spermatid over there. And this also has some mitochondria present in them. And also a centriole. A centriole is also present in them. Now, what happens is, as the Golgi complex goes and attaches on to the head region, an acrosome like head region is formed. And as this is forming and as the maturation is occurring over there, they start to contract in size. As a contraction occurs, the mitochondria that is present inside the spermatid goes and settles at the neck region over there. The mitochondria is getting attached onto the neck region. So this is the neck region over there of the sperm. The acrosome head is being formed. The Golgi apparatus is present at the head region. Mitochondria at the neck region. And as this is forming, the centriole that is present over there, that goes and forms a flagella-like structure or a tail-like structure over there, which helps in swimming of the sperm to reach the ovum. Now, as this is going on, there is some excess cytoplasm that is present. This is getting digested and reduced in number. And what you ultimately are left with as is a acrosome, the sperm with its head over there, the middle piece or the neck region over there, and that is the tail region over there. Now, this head region, the acrosome is going to have, remember, Golgi apparatus over there. Why Golgi apparatus? Golgi apparatus is necessary for production of some enzymes to digest the cell wall of the ovum. We'll talk about that during fertilization of this. So you have the Golgi apparatus at the acrosome head region, the nucleus, which is having 23 chromosomes and 22 autosome and one sex chromosome. That is, it can either have an X chromosome or a Y chromosome, and that will tell whether the child that is born is going to be a male or a female, depending on the sex chromosome that is present on the sperm over there. So a mother is going to give always an X chromosome. It is a father that is going to give whether is going to decide whether the child is going to be a male or a female, depending on the sex chromosome that is present in the sperm over there. So that is the maturation of the sperms over there. Now coming to oogenesis over here, that is development of the ovum or matured egg over here. The process of development and maturation of ovum is called as oogenesis. Genesis is to give rise to. Okay. Now, there are a pair of ovaries over there. So you have a pair of ovaries located on the lower abdominal cavity of the female over there. So lower abdominal cavity, there's a pair of ovary over here. The ovary is, again, there's a fallopian tube that is getting attached to the ovaries over here. So you have the uterus over there. From the uterus, you have a pair of ovaries coming from either side. And the ovary also has a fallopian tube, which is in contact with the ovary on either side. And this opens into the uterus and the cervix over there. Now, the ovary is a fibrous connective tissue called stroma. So this is made up of fibrous connective tissue called stroma. And they also have blood vessels and the lymph vessels connected. Of course, the nerves and the follicles in various stages of development. What is this follicles? Now, if you can see, this is a smaller follicle. This is a matured primary oocyte over there. This is developing oocyte over there. So various stages of the ovary is found over there, and this is present in the follicles over there, the follicles of various stages, and this is further divided into cortex, outer cortex, and middle medulla over there. So that is a medulla over there, that is a cortex over here. And of course, along with blood vessels and lymph vessels. Why blood vessels and lymph vessels? Because they need oxygen supply. Okay, so each time during each ovulation, whenever the egg has to be produced over there, one follicular cell develops to become an ovum. So you have the ovary over there. In this ovary, you have this follicle cells. Now, this follicle cells, one of them will be decided which one will become a matured egg over there. Now, this develops, grows in size to form the matured egg over there. So the follicular cell will decide which one has to grow over here. So, for example, if you can see in this particular picture over here, so that is a primordial follicle. Primordial follicle will have to undergo the proliferative stage to form primary follicle. Primary becomes secondary follicle. That is, they grow in size, and that becomes a primary oocyte, and then primary becomes secondary oocyte, so on and so forth. How the development occurs, we'll be talking about that. Now, once the follicle, you find a mature follicle over this. This is a mature follicle. Now, this mature follicle is called as Graafian follicle, because Graafian means they are matured enough, they are big enough, and they are ready to be ovulated. That is called as Graafian follicle. The biggest one is called as a Graafian follicle. Now, as this going on, when the primary, the follicle is growing in size, the egg, the nucleus is covered by zona pellucida first. So this is a thick layer of zona pellucida. Around the zona pellucida is zona radiata. This is a thick follicle cells that is covering the primary oocyte over there, the zona radiata and the zona pellucida. Now, why am I'm stressing these two areas over there? Because the sperm has to digest this zona radiata and zona pellucida to get itself inserted into this egg over here. So that is the reason why these two layers are very, very important, zona pellucida and zona radiata. And as this is growing, there is vacuum that is created that is called as antrum over there as the graphene follicles is increasing in size over there. Thick interna and externa over here, that is the haploid secondary oocyte. Haploid secondary oocyte. Why secondary oocyte? You are having only 23 from 46 from here. Now you have only 23. And what is the sex chromosome that is found? X chromosome. It is going to only have X chromosome over there. So that's a mature secondary oocyte that is present over here. Okay. Now, as this is going on, there are three key stages that is occurring over here for the process of oogenesis, the same way what we talked about in the spermatogenesis, multiplication phase, growth phase, and maturation phase. Now, in a multiplicative or proliferative phase, now remember there is a little bit confusion over here and a little bit difference in the growth from spermatogenesis and oogenesis. What is that is happening over here? So when the fetus is forming again, as a fetus is forming from the yolk sac, the cells that moves to the gonadal ridge or genital ridge is called as the future gonads. Now this is called as the primordial germ cells that goes and settles itself in the gonadic region. Now as the cells are going to grow, what is happening is there are maximum number of cells that are present over here, which is going to form this primordial germ cells. Now, the primordial germ cells get differentiated within the ovaries to form oogonia. And this oogonia, which is a diploid number, that has to divide mitotically to produce 7 million germ cells. Now, mind you, the 7 million germ cells are being developed when the fetus is still forming in the mother. That is, when the fetus is 4th to 5th month, you have 6 to 7 million oogonium. So, for example, if you think that this is a 5th month or a 4th month or a 5th month fetus over here, now this is going to have 7 million primordial cells, the germinal cells that are present in there. Now, all the 7 million germ cells are not going to stay for long. Now, as after this is formed over there, now as the next trimester comes over here, now almost the cells that are present over here will undergo atresia. Atresia is called as cell death over there. Now, this 7 million oogonium that are present in the germinal cells will die, and only 1 million germ cells remain over there. Imagine if all the 6 million germ cells are going to be there, then definitely the ovary size is going to be bigger enough. So you don't have that much. So atresia occurs over there. All the 7 million germ cells will have to undergo cell death over this. And what is left with only by the time the child is 9th month, when the fetus is 9th month, just before the birth, they are having only 1 million primary oocytes over there. That is proliferative phase over there. Now, growth phase. What is happening in the growth phase? Now, as the fetus is growing in the mother, the primary oocyte have started to undergo meiosis 1 or meiosis 1. The primary oocyte is present over there. That is, primordial germ cells have become proliferated to form primary oocyte. Now, this primary oocyte, which is going to be formed, this will start to undergo meiosis 1 over there. Okay, so meiosis 1 is going on. Mind you, still before birth, that is, as a fetus is developing in the mother, the meiosis 1 has started in the fetus's ovary. Okay, so that's why they say when a mother is pregnant with a girl baby, she's already a grandmother. Why? Because the ovary has started to do its work over there within the fetus over there. So here you have the primary oocyte, which is undergoing meiosis 1. Now, what is happening over here? The meiosis 1 gets arrested at prophase 1. Now, what is prophase 1? That is, all the chromosomes, the parent's chromosome has undergone crossover and exchange of information has occurred, and they get arrested at that point over there. Now, the child is born. Now, as a child is born, it is going to stay in that arrested form till the child matures, till the baby girl matures at age 13, 12 or 13. When they mature, when the puberty sets in, then meiosis resumes. Meiosis 1 resumes, and they get arrested at meiosis 2, where at metaphase stage. So meiosis 1 has resumed. Meiosis 1 has completed. It goes into meiosis 2. And in the meiosis 2, they get arrested at metaphase 2, right? So at the metaphase 2, it has got arrested. Complete meiosis has not occurred. The completion of meiosis 2 occurs only when the egg is fertilized. Otherwise, it stays as such in meiosis 2 metaphase stage over there, right? So approximately at birth, you have left with only 1 to 1.5 million primary oocytes. That is what we talked about here. 1.5, to 1.5 primary oocytes remain, and out of which only 35,000 are present by the time the child attains puberty. Why? Because those cells that are present in the ovary are dying over there and some are aging over there. So you are left with only 35,000. And during women's reproductive lifetime, only 300 to 400 oocytes mature and ovulate over there. Ovulation is releasing of the egg occurs. And so the whole lifetime, the lady is able to give rise to 300 to 400 oocytes, mature oocytes and ovulate. Remaining primary oocyte will undergo atresia. Atresia is cell death. So this is a clear picture to understand what I was talking about. The diploid oogonium before birth, that is in the 3rd month to 7th month of gestation, the fetus is going to produce primary oocyte. The primary oocyte undergoes meiosis 1. The meiosis 1 gets arrested at prophase stage, prophase 1 of meiosis 1 stage. Now, after the child birth is occurring and after the child has grown and attained puberty, once in the puberty, the meiosis 1 resumes and it completes meiosis and secondary oocyte is formed. The secondary oocyte undergoes meiosis 2. The meiosis 2 is arrested at a metaphase stage over there. So meiosis 2 gets arrested at the metaphase stage 2 over there. And the meiosis resumes only when the egg is fertilized. Otherwise, the ovulated egg, which is still in a metaphase 2 stage over there, will be removed if the egg is not fertilized. And during division over there, during meiosis 1, when the primary oocyte becomes secondary oocyte, it gives rise to one secondary oocyte and one polar body. Now, what is the difference over here in the polar body over there? One egg is larger and one egg is smaller over there. Oocyte is smaller over there. The smaller oocyte is called as first polar body, and the bigger one is called as a secondary oocyte. Now, why this partial division of one is big and one is small is because the egg has to store in lots of cytoplasm, lots of cell organelles for the development of the zygote over there. And that is the reason why one egg takes in all the maximum amount of cytoplasm and cell organelles present in them. Then the second one, the first polar body that is formed is having very less amount of cytoplasm, and this eventually cannot be fertilized because they are very small and they cannot form into a zygote over there. Now, if this remains in the system, then this will also undergo meiosis 2 over there to produce the secondary polar body over there, two other polar bodies, but this is going to be dead over there. The one that is going to become secondary oocyte will undergo secondary meiosis over there, the second meiotic division over there. Now, this second meiotic division will give rise to one fertilized egg, one matured egg, and other secondary polar body over there. Now, this also gets degenerated. Totally, you have three polar bodies. Now, this will divide into two, and this has one. So, 2 plus 1 over there, 3. 3 polar bodies is dead. The one that is going to stay is a mature ovum, which is having lots of cytoplasm. Now, why I said it has to have lots of cytoplasm is they have to give some energy for the zygote to undergo cleavage over there, which is going to get fertilized over there. So that is the picture of oogenesis over here. So maturation phase, I've already talked about maturation phase over here. What is exactly a maturation phase? Now, when the secondary oocyte is ready over here, the secondary oocyte, which is arrested at the metaphase 2, undergoes ovulation. That is, they have to be released from the ovary. Now, this release of the ovum from the ovary is called is helped by luteinizing hormone. So you have these hormones that are being given from the hypothalamus of the head region. From the brain, hypothalamus releases some hormones, and the maximum amount of hormone, that is luteinizing hormone, speeds up when an egg is getting matured and when they are ready to be released. Luteinizing hormones is released. That will help the secondary oocyte to rupture and releasing the ovum over there, matured egg over there. And the rest of the body is called as corpus luteum. Now, this corpus luteum that is empty without the egg, the ovum inside them. Now, this is the one that is going to produce maximum amount of progesterone for the development of the zygote in case the egg is going to get fertilized over there. Okay, so the release of the oocyte from the ovary marks the transition from puberty to reproductive maturity in the females, and throughout reproductive year, ovulation occurs approximately every 14th to 15th day. That is, when there is a 28-day menstrual cycle, on the 14th or the 15th day, ovulation occurs. And if the released ovum is fertilized, then there is no menstrual cycle in the next cycle. But if the egg is not fertilized, then the egg is going to be removed out of the uterus and the menstrual cycle will resume as 28-day cycle over there. So this is the ovulation of the ovulation and the hormones over here. So what is these hormones that are important for this ovulation to occur? So from the hypothalamus, from the brain, hypothalamus is going to release gonadotrophic releasing hormones. And this gonadotrophic releasing hormone is going to stimulate the anterior pituitary gland. Anterior pituitary gland, once stimulated by this gonadotrophic hormone, is going to release gonadotropins. Now, the gonadotropins is the one that is going to release follicle stimulating hormones and the luteinizing hormone. The follicle stimulating hormones during puberty starts to develop the the primordial germ cells to primary oocyte, and this primary oocyte will become secondary oocyte over there. And once it is becoming secondary oocyte, the luteinizing hormones increases, and the luteinizing hormones is the one that induces ovulation to occur. So from here till here, follicle stimulating hormones are activated, and this part, luteinizing hormones are activated to release the ovum from the secondary, the follicle, Graafian follicle over here. So that, what is a Graafian follicle? This is a highly developed follicle called as a Graafian follicle. So from the Graafian follicle, the ovum has to be released, and this is taken care of by the luteinizing hormones. And this, after removal of the ovum, the Graafian follicle forms into corpus luteum. And this corpus luteum is the one that is going to give signals to the brain again to produce more amount of progesterone and estrogen. So this is the estrogen and progesterone that is going to be released. That will take care of the placenta, the endometrium in the uterus for the induction of the fertilized egg over here. So what is happening after the ovulation has occurred over here? So here, when you see ovulation over here, after ovulation has occurred, you can see over here, after the ovulation has occurred over here, once the egg is released from the ovary, the fallopian tube is the one that is going to take the egg, the fimbriae or finger-like projections that are present over here. So the fallopian tube is going to take the egg that has been released from the ovary. And over here, if the sperms are present over here, the sperm is going to fertilize the egg over there. And once the sperm is going to enter by breaking open the zona pellucida and zona radiata. Remember, the wall that is present around the matured egg is the zona pellucida and the zona radiata. It is not easy for the sperm to easily enter them, but only one sperm is able to enter with the help of the enzyme that is present, that is going to be released by the Golgi complex. Remember, the Golgi complex is going to release some digestive juices. That juices is going to cut open the zona radiata and then the zona pellucida. It is not going to completely go and attach to the nucleus over here, because this nucleus is the egg nucleus over there. Once this is attached over there, then meiosis 2 resumes. Remember, I told you all meiosis 2 metaphase stage, it has stopped over there. Now, once the sperm is going to fertilize the egg, then the meiosis 2 resumes over here. From the metaphase, it goes to anaphase, telophase by producing one secondary polar body and a matured ovum. And this matured ovum with its 23 eggs is going to get fertilized with the nucleus of the sperm that is having either 23 eggs or 23 Y to give a child, a girl or a baby over there. So what is happening over here as this is occurring over there? I just want you all to see the stages that is occurring inside the uterus over there. What is happening over there? So, for example, if you're going to take this 28-day cycle, on day 1, if the menses has occurred over there, That means there is no, the ovary is still and the developing stages over there. The endometrium is going to be cleaned off, or endometrium is going to be removed from here. Now, this is the growth of the endometrium, as you can see over here. So, day 0, menses has occurred. Now, after that, what happens is the follicle stimulating hormones is increased, and as the follicle stimulating hormones is increasing, one of the follicle cell is going to be selected, and they start to grow into primary oocyte. Primary oocyte is going to become secondary oocyte. Now, as the Graafian follicle is increased, luteinizing hormone is going to be increased over there. Now, as this is going on, the endometrium is getting developed over there. The walls of the uterus is getting developed over there by the endometrium cells over there. Why? Because the endometrium is the one that is going to incorporate the fertilized egg over there. So they need to have a bed-like structure for incorporation over there. And when the luteinizing hormone has peaked up, ovulation occurs, and once the egg is being released, the egg is taken care of from the fallopian tube. They move into the pipe, and somewhere over here, if the sperms are present, the egg is fertilized, they move and they get incorporated into the uterus of there. If the egg is not fertilized, they move and they get washed away over there. The incorporation doesn't occur. Why? Because the egg has not completed its meiosis 2 over there. So that is how the body understands whether this egg is fertilized or not fertilized over there by the proteins that are present around the structure of that fertilized egg. Okay. So if the uterus finds out that the egg is fertilized, then the endometrium is not sloughed off and the egg is going to get incorporated, and the progesterone stays put over there to take care of the development of the fetus over there. The zygote is going to get developed over there. So when you're talking about this fertilization, as I told over there, the fertilization, the sperms comes over here. You have the zona pellucida and the zona radiata. The acrosome gets attached to the zona pellucida layer over here. And the hydrolytic enzymes that are present in the Golgi complex is going to digest the zona pellucida. And once inside, the zona pellucida is completely closed, and this is not going to allow the second sperm to enter inside. One egg, one sperm. No two sperms are going to enter inside this egg over there. And once inside over there, once this is incorporated, the cell will complete its meiosis 2. And once the meiosis 2 has occurred, the sperm nucleus goes inside and the fertilization occurs. That is the fertilization of the egg over there. Yeah, so this is what I was talking about. And on day 9, the implantation starts to occur. That is the egg after fertilization. Implantation occurs. And during the fertilization over here, on day 2 itself, the cell starts to divide itself. The cell has to divide itself. That is, cleavage occurs over here. And during the cleavage is occurring, they get incorporated in that particular placenta, the uterus over there. So coming to types of eggs over here. Now, as we have talked about the fertilization over here, there are different types of eggs that are found in the nature over here. Now, when you're talking, you might also be asked to discuss about different types of eggs. You might be asked for 5 marks also, short answer over here. Now, when they're asking you for types of eggs, you are going to talk about different types of eggs over here. They are divided into alecytal, microlecytal, mesolacytal, and telolacytal over here. Meso and telolacytal is also called as megalecytal. Alecytal, A means absent. Lecytal means the amount of yolk that is present. A is absence of yolk is called as alecytal egg. Micro is there are very few amount of yolk that is present over there. That is the nucleus, pronucleus over here, and the blue color one is the cytoplasm over there. So less amount of yolk is called as microlecytal. Mesolacytal is medium amount of yolk is present in the egg, that is called as mesolacytal. That is the cytoplasm, and that is the nucleus. The nucleus is the chromosomes that are present over here. And megalecytal is this large amount of yolk that is being present over there, the nucleus, and a little bit of cytoplasm is present over here. Mega means more amount of yolk. So these are the types of eggs that is you'll have to discuss. And this types of egg is based on amount of yolk, distribution of yolk, presence and absence of shell, that is cleidoid and acleidoid cells, and pattern of development. So alecytal, I've already talked about this, less amount or no yolk at all. Example is mammals and human eggs. Why human eggs have less amount of yolk? Because in the humans we have this placenta that is present, and the cord, umbilical cord that is present, where the mother is directly feeding the developing embryo inside. So that is the reason why the human and the mammals do not need to have the yolk over there in the eggs. It's only the cytoplasm and the nucleus that is present over there. Coming to lecithin eggs over there, that is amount of yolk that is present, divided again into micro, meso, macro. So the microlecithin, very less amount of yolk, is found in examples, echinoderms, urochordates, and amphioxus. Mesolecithin, medium amount of yolk, examples are mollusca, amphibians, dipnoi, and petromyzon. And macrolecithin, or more amount of yolk, examples are bony fishes, reptiles, birds, egg-laying mammals over there. Now, according to the distribution of the yolk, you can see the distribution of yolk, you can find different types over here. So according to that, they can be of three types: homolecithin, heterolecithin. Homolecithin, or also called as isolecithin over there, the yolk is uniformly distributed inside the egg. So yolk, you can see it is uniformly distributed around the nucleus, and the center part is the cytoplasm over there. And the examples of here is again echinoderms, urochordates, and amphioxus. And heterolectal eggs, these are again of a little bit of different patterns over here. That is telolecithin. Under telolecithin, you will have high amount of yolk is present on the vegetal pole, and at the animal pole, you have the nucleus over there, and the cytoplasm is very much less over there, pushed towards the nucleus over there. That is the telolectal eggs. The centrolecithin is where this telolectin is of two types over there. Here you have where maximum amount of yolk and less amount of cytoplasm pushed to one side, and here you have yolk on one side, a little bit of yolk on one side, the nucleus over there on top, and the cytoplasm is around the yolk and the nucleus. This is one type of telolectal eggs you find over there. And the centrolecithin is where the yolk is present in the center. This is different from the homolectal. The homolectal yolk is outside. In central lecithin, the yolk is in the center, surrounded by the cytoplasm and the nucleus in the center. So mostly insect eggs have this type. And these two type of eggs you find in fishes, amphibians, reptiles, and birds. That is the telolectal over there. Now, coming to cleavage pattern, this is one of the most important topics under embryology, and also one of the common questions that is being asked for the examination point of view for cleavages over here. Here will be, I'll be going a little bit fast over here. If you have any doubt, you can keep contacting me by, or you can also ask your counselors in the college for further details over here. But here we'll talk about cleavages. What is cleavage over here? Now I said once the ovum is produced, once the ovum is fertilized, they have to after fertilization they become zygote. Now this zygote has to undergo cleavage over there from single cell to two cell stage, from two cells to four cell, four to eight, so on and so forth over there. That is called as cleavage over there. Now this cleavage has a pattern. Not all eggs will, zygote will undergo the same pattern of cleavages. That is why we are going to talk about the patterns over here. So this can be either spiral, radial, or bilateral type of a cleavage over there. So we'll talk about this cleavage systems over there. Now, where we are talking, what are we talking about? We talked about the sperm and the eggs. The sperm and the eggs, fertilization has occurred. The fertilization has given rise to zygote. Now, this zygote has to undergo cleavages. That is, mitosis has to occur for the cell division to occur over here. So here, what is happening over here under the mitosis activity over there? The repeated mitotic divisions will give rise to different cell stages over here. So this cleavage is a series of mitotic division whereby enormous volume of egg cytoplasm is divided into smaller nucleated cells. So here what I'm talking about, the cytoplasm and the nucleus is going to get divided over there, not the yolk. We are only talking about the nucleus and its cytoplasm here, which is that one you can see. Now, yeah, you can see this one. This nucleus and the cytoplasm has to be undergoing the divisions over there. Equal distribution has to occur over here. So what is happening over here? As the division is going to occur, you have to remember one thing. the cell that is of the size is not growing in size, but within its structure, the division is occurring, whereby the cytoplasm is repeatedly getting divided to each daughter cells. So they are not increasing in volume, but they are only getting divided within itself without itself becoming big over there. So whatever the amount of cytoplasm is present, that itself is getting shared into each cells over there. So here you can see into each cell, the amount of cytoplasm is going to get divided. And this division of cells is called as blastomers. Blastomers is a name that is going to be given for the division. And you have a small gray crescent over there. The gray crescent is where the fertilization, the sperm entry has occurred, and so the name gray crescent over there. So that is what it says. During cleavage, however, the cytoplasmic volume does not increase. Rather, the volume of the zygote cytoplasm is divided into increasingly smaller cells over there. That is called division. And these cells are called as blastomers. These cells are called as blastomers over there. So you can see the size is the same from this point to this point. The size is the same, and they have not grown in size over here. Those are called as blastomers. So this decrease in the ratio is crucial in timing the activation of certain genes. What is the decrease of here? The decrease of the cytoplasm is crucial in timing the activation of certain cells where it has to activate it to start moving from its place over there. So the genes have to get activated. The DNA have to get the signals so that the cell starts to migrate from one place to another place. That is called as the decrease in the ratio is crucial for timing the activation of certain genes over here. So, for example, in frog Xenopus levis, transcription of new messages is not activated until 12 division. So this is first division, second division, third division. So until 12 divisions, they are not activated. After the 12th division, then the blastomers becomes motile. What is motile? They can move. The movement occurs over there in these cells over here. That is, they have to move from one place to another. Why? Because each cell that has been divided is destined to form one part of a tissue, and that tissue becomes an organ. So this destination starts, the movement starts only after 12th division, in which one? In Xenopus, that is a frog over there. So each species has its own different timings over there. And this stage is called mid-blastula transition. What is mid-blastula? From the blastula, that is the blastomers, it goes into the next stage called gastrulation, and this is called as MBT stage or mid-blastula transition over there. So here you can very clearly see this is one cell with a nucleus, and this has divided with two nucleuses, four cells with four nucleus, and so on and so forth. Divisions that is occurring over here. So rapid cell division, that is, so from zygote, the cleavage into 8-cell stage. From the 8-cell stage, they form into blastula stage. The blastula has a blastocele in the center. When you cut open the egg, you can have a vacuole in the center, that is the cavity that is present in the center, that is called as a blastocele. The upper part of the cells are smaller, and as you go down, the larger in size over there. That is again a destination of the cells over there that is destined to become one type of a cell. Now this goes into the next phase called the gastrulation. Now this phase is called as the MBT phase. What is MBT over here? Mid-blastula transition. So from blastula stage, when it goes to the gastrulation, that is called as MBT stage. And this gastrulation occurs so that it gives rise to three important germ layers, that is the ectoderm, endoderm, and the mesodermal germ layers. Now, before that, we'll be talking about this cleavages, how the cleavages are occurring, and what are the shapes that you find in this cleavages. So for that, you'll have to understand the plane of cleavages over here. The plane of cleavages of four types. The first one is a meridial plane of cleavage over there. If the egg is, if the zygote is divided from animal pole to the vegetal pole, now this is called as meridian plane of cleavage over there, or the cutting the egg into two equal cells. Now, there's one more called the vertical plane of cleavage. In the vertical plane of cleavage, the cleavage occurs between, on the either side of the animal, the meridian plane. This is a meridian plane. On either side, if there is a cleavage that is going to occur, then that is called as a vertical plane cleavage. This vertical plane cleavage will give rise to unequal size of the division that is going to be formed. Then the third one is the equatorial type of a cleavage, where the equatorial plane is going to divide the egg into two equal halves over there. So that is equatorial plane. And the last one is a latitudinal plane of cleavage, or the transverse plane of cleavage. And this transverse plane of cleavage will occur on either side of the equatorial plane. And this will also give rise to unequal part of the division that is going to occur over here. So this is the transverse plane or latitudinal plane of cleavage. Now coming to pattern of embryonic cleavage over there. The pattern of embryonic cleavage is different in different types of the species, as I told you. In So, for example, I want you all to understand this part over there. What is an animal pole and what is a vegetal pole? Animal pole is where the nucleus is present, that is called as animal pole, and the place where the yolk is present, that is called as a vegetal pole. Why vegetal pole? Vegetal pole is nothing but the food that is going to be provided for the development of the animal over there. So that is why the nucleus is called the animal pole. Wherever the nucleus is, that is called as the animal pole, and wherever the yolk is, that is called as the vegetal pole over there. Now, pattern of embryonic cleavage over here, the primary chordates division is even over there, and towards a symmetrical blastula composed of equal sizes. You can find the equal sizes over here. So in mammals, it is a holoblastic cleavage, that creating a trophoblast containing a blastocele. So that's a blastocele over there. Now, holoblastic cleavage will give rise to blastocele over there in the mammals over there. And in reptiles and birds, it's a meroblastic cleavage which occurs, resulting in a caplet structure. So meroblastic cleavages, so for example, you have this nucleus over there, and you have the yolk, maximum amount of yolk that is present in the vegetal pole. The division is occurring only in the zygote over here, wherever the nucleus is. So you have this division occurring only in one part and surrounded by the yolk over there. And as the division is going to occur, this will create a caplet structure. So that is what this cleavage is called as a meroblastic cleavage, resulting in a caplet structure on top of the yolk. And this is an incomplete cleavage over here. In the holoblastic cleavage, it is a complete cleavage, resulting in a blastocele over here. And in amphibians, holoblastic cleavage results in a asymmetrical blastula. So you have a asymmetrical blastula over there. So you don't have a proper divisions of the cells over here. And this is a blastocele, very small blastocele that is present. And this holoblastic cleavage over here, even though it is holoblastic over here, on the eighth cell, instead of having two equal sides of the cells, the top layer is small cells and the bottom ones are larger cells over here. And that is the reason you have an asymmetrical blastula cleavage. That is a pattern of the holoblastic with a blastocele. The reptiles having a meroblastic with a caplet structure. Amphibians also holoblastic but asymmetrical blastula is found in this type of cleavages over there. Now, before going into a little bit detail, I just want you all to understand the shape of the egg over here. I don't have many students over here, so I'll just give you all this answer over here. Everyone must have seen this egg over there, the eggshell over there, the eggshell covered with the albumin, that is the white color, the albumin. And the albumin is going to have the chalaza over there, the white color part. When you break open the egg, you might have seen this white color thread-type structures on the either side of the yolk over there. That's called as a chalaza. And this chalaza will keep the yolk in position, doesn't allow them to move from one place to another over there. And then it also has a vitelline membrane around the yolk over there, which keeps the yolk in position. Otherwise, what would have happened if there is no vitelline membrane? The whole yolk must have got mixed up with the albumin over there. Along with that, you have a air cell over there, air gap over there, where the oxygen and carbon dioxide exchange occurs from this gap over there. If you boil an egg, when you open it, you can clearly find that the space over there, that is the air space or the air cell over there. And the yolk is also going to have a germinal disc. I'm sure whenever you must have broken the egg over there, you must have seen a white dot on the yolk. And this white dot is nothing but the germinal disc, which is having the nucleus. And which part of the egg is going to become the chick over there. Most of the students answer that the yolk is the one that is going to become the chick, and some say the albumin part. Now, this part, that is the germinal disc that is present, that white spot over there. Now, that is the spot where you have the nucleus, and that is the spot which is going to become the young chick. Now, this yolk is nothing but the food for the development of the chick over there. So, yolk is not the chicken, but the egg, the germinal disc that is present is going to form the chick. Why the chick is yellow in color? Because the yolk is yellow in color. The food that it is taking will be yellow in color. So, the pigment is yellow. But as they grow, the pigment changes. The melanin sets in, and we have a different colors of the hen over there. But when young, it is yellow. And so this is the one that I want you all to remember. The germinal disc is the one that is going to become the young chick over there. Now, coming to the influence of yolk on the cleavage over there, the amount of yolk and its distribution will affect the process of cleavage over here. So we'll talk a little bit about holoblastic and meroblastic over here. So when in the holoblastic cleavage, that is in the placental mammals and amphioxus, equal holoblastic cleavage occurs. That is in microlecital and isolecital. What is micro and isolecital over there? Micro is very, very small amount, alecital and microlecital, very, very less amount or no amount of yolk is present. In these type of eggs, you have this holoblastic cleavage. So you have a complete cleavage from animal pole to vegetal pole or north pole to south pole over there, dividing the zygote into two equal halves. Now, this two equal halves will undergo another type of a cleavage over there on the either side, opposite to the one that has occurred over there, dividing the two cells into four. Now, there is one radial cleavage that is going to occur over there, the cleavage that is going to divide these four cells into eight cells over there. Now, these eight cells are again equal in size. I want you all to see that the third division that is going to occur is going to divide the eggs into equal sizes over here. So, that is the equal holoblastic cleavage. And unequal holoblastic cleavage over here, when you're talking about unequal holoblastic cleavage, the third division that is going to occur, this is going to occur a little bit on the top. So if it is going to occur over here exactly, then you're going to have something like this. But the third division is going to divide the cell into unequal. That is, the upper cells are going to be smaller one over there, and the lower cells are going to be macromeres. That is a micromeres and the macromeres, the larger cell over there. So that is called as unequal holoblastic cleavage that you find in the amphibians and other cells, other species over there. Now, what is a meroblastic cleavage? Meroblastic cleavage is mostly found in the reptiles and the birds. Meroblastic, where you have a cap-like structure. I told you already the pattern of cleavage over here, the discoidal cleavage over here. So you have the yolk over there because of the presence of maximum amount of yolk over there. The nucleus and the cytoplasm is pushed to one side on the top over there, and the division starts to occur on above the yolk over there. So that's the yolk, and this is the nucleus. The division occurs over there. As the division keeps on occurring, there's a cap-like structure that is formed. So that is called as meroblastic cleavage, the discoidal cleavage over there. And this is a discoidal type of a cleavage. Now, when you're talking about the superficial cleavage, what's the difference between this cell and this cell? It's nothing but this is magnified. This particular picture is magnified and shown on the lateral side of how they look like over there. If you're going to see a bird's view from top, this is how the division is going to occur. So this is something like this over there, that is a cap-like structure that is being formed on the neuroplastic cleavage. What is a superficial cleavage? Superficial cleavage you find in centrolecital type of eggs over there. Centrolecital type of eggs you find only in the insects over here, where the yolk is present in the center and surrounded by the cytoplasm and the nucleus in the center. So what's happening over here now, because the cytoplasm is far away from the yolk over there, now division starts only in the nucleus. As the nucleus is getting divided, the cytoplasm is not getting divided alongside the nucleus over there. So first the nucleus is getting divided. As the nucleus is getting divided, and when they start to scatter around, they move towards the cytoplasm over there. Once they move towards the cytoplasm also starts to divide over there along with the nucleus over there. the cytoplasm goes and surrounds each nucleus over there. So that is called a superficial cleavage, which is found only in the insects over there. So that is one type of cleavage. Why? Because this is the centrolecytal yolk is in between the cytoplasm and the nucleus, and that is the reason why first the nucleus divides, they move around, and once they move out towards the cytoplasm starts to divide. That is superficial type of a cleavage over there. And coming to the pattern of cleavage, I'll take another 5 minutes over here and complete this topic. Pattern of cleavage is different types over here. The first one is a radial type of a cleavage. Under the radial type of a cleavage, you have one division over there. That is, in this regular cleavage, division is at right angles to the earlier one. So once you have one straight horizontal, the vertical type of a cleavage, then the next one is horizontal over there. So that is right angle to the earlier cleavage over there. The cells are placed just one above the other, and the upper 4 blastomeres are arranged just above the lower 4 blastomeres. So this is a radial type of a cleavage over there. And the spiral type of a cleavage, spirally they get cut around. Something like you have this potato that has been cut spirally. So spiral type of a cleavage can occur in annelids over here. So you have the planes are oblique in structure, so they are obliquely cut. 4 blastomeres of the lower plane rotate clockwise, and the rotation was also different over here. If the rotation is onto the clockwise direction, then this is called as dextral spiral cleavage found in mollusca. And if this is anti-clockwise direction, if this rotation is anti-clockwise direction, then this type of cleavage is called as sinister spiral cleavage found in platyhelminthes and annelids over there. This is one type of cleavage. Then the next one is bilateral cleavage over here. So what is a bilateral cleavage over here? Due to unequal holoblastic cleavage, bilateral symmetry is established, and the blastomere of one lateral side are smaller in size and another 4 lateral sides are larger in size. So if you can see over here, this is divided into 2 cells. Now the next cell, there is a division of unequal sizes over here. So you have on either side, the opposite side is a larger one and the other sides are smaller one over there. And as the division goes on, there is a difference over here. So you can see over here, first quarter, larger one, they are divided into 8 cells over there. And these 8 cells are larger, then the next cells are smaller, and then the macromeres are formed. So that is the biradial cleavage of different types over there. And rotational cleavage, something like the spiral cleavage over here. This rotational cleavage is found in humans over there. So how is this rotational cleavage is occurring over here? So you have this first cleavage that has occurred over here. Now the next cleavage, one cell will undergo lateral type of horizontal cleavage, and one will go vertical type of a cleavage over here. So here you can see this has undergone the horizontal type of a cleavage to give these two, and this will undergo lateral. This is horizontal and this is lateral type of a cleavage. So each one is going to have different types of cleavages over here. One is having a lateral, one is horizontal, one is lateral, one is horizontal. That is 90 degrees from each other is going to occur. And this is because mammals have very less amount of yolk, and hence they have to undergo this type of a cleavage that is called as a rotational cleavage over here. So you can see the plane of cleavages that is going to occur over here. If one is going to be cleaved like that, the one is going to be cleaved the other way around over there. Vertical and horizontal type of cleavage occurs over there. So this is about the cleavages that we saw over there. Now you have to remember why cleavages are necessary. Cleavages are necessary so that there are a number of cells over there from blastula, they have to become gastrulation stage over there. The 8 cell stage becomes morula stage, and the morula stage becomes blastulation. Blastocele is formed, blastula 2. The next stage of development is called as gastrulation stage. For this to occur, the cell have to undergo this cleavages over there. So the cleavages are different in different type of species over there. So what are the cleavages that we saw? We saw the rotational cleavage, the biradial cleavage over there. Then we saw the spiral cleavage, the radial cleavages, and these are the type of cleavages that we saw. Now, this cells after cleavage will undergo MBT stage. MBT, mid-blastular transition. From mid-blastular transition, they have to go to gastrulation stage. This gastrulation stage we will continue in the next class, that is on next Sunday. where we'll complete this gas relation and also new relation. And I'll also try to complete a little bit about the limb development also. Okay, so I hope you have understood this topic over there. As again, as I said, if you have any doubts, you can consult counselors over there who will be able to help you all to understand this topic. Good evening, students.